Hi guys, I wanted to take a few minutes today to do a little lesson on mitosis and also cancer. So mitosis, you should have learned about yesterday, is also referred to as cell division or sometimes the cell cycle. You'll kind of see those words used interchangeably. But what mitosis is it's how cells are copied. It's like the copy machine for cells. And so the reason that cells need to be copied, well, there are a few. So first for growth, okay? So living organisms need to grow, right? We all began as one cell. When sperm meets egg, it makes that first cell of an organism called a zygote. And we all began there with one cell. But now we're made up of millions of cells, or maybe even billions of cells. And so how did we get there? Well, we got there through mitosis. That one cell divided and it made two cells. And then those two cells divided and made more cells and more and more. And as you continue to grow, your cells don't just get bigger, you get more cells. And that happens through mitosis. So mitosis helps and allows organisms to grow. Now, you may think, well, Miss Brown, you're grown now, you're an adult. Are you still doing mitosis? Yes, I am. And that's because mitosis is also for repair and replacement, okay? So skin cells, for example, your skin cells are constantly sloughing off, right? And so you are constantly replacing cells. Skin cells are replaced constantly. You're constantly making new ones. Some cells, like skin cells, are replaced very frequently. Other cells, like red blood cells, are replaced not as frequently. Red blood cells usually have a lifespan of like 3 to 4 months. Some cells, like nerve cells in your brain, those are not replaced. So if you kill them or damage them, they don't grow back. But mitosis is for replacement and repair, okay? Some organs, like liver tissue, can grow back. And so it can be repaired if it is damaged. And so mitosis helps to do that. So in mitosis or in the cell cycle or cell division, there are major steps. And I don't remember exactly if PLTW talks about all of these, but I'm going to because this is the whole shebang, okay? So technically, mitosis or cell division is just this part. The PMAT is the mitosis cell division part. The rest of it is the whole cell cycle, okay? So in PMAT is where the division happens, but you have to do some things before and after that division to make it happen. So I'm going to walk through those stages. Now, this is a diagram that I use with AP Biology, and so we're not going to touch on all of these terms because you don't need to use it that deep, but I really liked the pictures. So don't worry about, all of the words so much because you're not going to be responsible for those, but just maybe take the notes on what I talk about or, update from what you did yesterday, add to those diagrams. So interphase is the step that precedes mitosis where the DNA is copied, okay? In mitosis, you take one cell called the parent cell and you break it in half and you make two identical daughter cells, okay? Well, that parent cell contains one complete set of DNA. So in order for both daughter cells to be identical and both also contain one complete set of DNA, then it has to go through DNA replication, and that happens in interphase. Now in biology, we talk about several different steps of interphase, but here we're just going to talk about interphase in general. And so interphase is where the DNA is copied. Notice also in interphase, you don't see chromosomes yet. This is the DNA, but it exists in all this. I like to think of it as it looks like dental floss pulled out of the tube or the container, right? But then when you condense that dental floss and you wind it into a shape, then you get these chromosomes. that we see. But it contains this chromatin, this non-condensed chromosome version of the DNA. But it's copied there in that picture, okay? So that's the I interphase. The next phase, which actually starts mitosis or cell division, is prophase, and that's our P. So in prophase, what happens, the main thing that you start to see here is the nucleus starts to break down, okay? So nucleus breakdown, N-U-C-L-E-U-S. You see chromosomes start to form. So they don't look exactly perfect, but they are starting to form. And you also see this, which is the spindle fiber forms. The spindle fiber is really important because it helps to separate the chromosomes or separate the DNA in the two new cells. So that's prophase, okay? We're not going to talk about prometaphase, that's getting really in depth there, but that's just kind of a later portion of prophase before you get to metaphase, okay? So we would still consider that to be prophase too. You notice that in here, the chromosomes are a little bit more condensed and the spindles are a little bit more formed. So that's still in prophase. Also, the nucleus here, that's what these purple things are continuing to be a little bit more further in the breakdown process, okay? Now, in metaphase, metaphases are M here. That's the next step. That is when they line up in the middle. Metaphase, M equals middle, okay? And you see that. All of the chromosomes are lined up here in the middle. You also see the spindle fibers have attached to the chromosome. That portion of the chromosome where they attach, you should have drawn, maybe you didn't draw a picture of this yesterday actually, is called the centromere. But the spindle fibers have to attach to the centromere in metaphase. And that's how in anaphase, or A, in the PMAT, I'll write that back up here, PMAT C, is where they pull apart. And you can always remember anaphase and apart. So those chromosomes are being pulled apart. All right? And then in telophase, you start to see the nucleus come back together. The chromosomes are starting to lengthen out because they're going to turn back into that chromatin. The spindle fibers have been reabsorbed. And then the last stage is cytokinesis, and that's the division of the cytoplasm. Cytokinesis in the division of the cytoplasm. And this picture doesn't fully show cytokinesis, but in cytokinesis is where you divide this. This is really telophase. In cytokinesis, you would see two complete new cells with fully intact nuclei. Okay? And they're identical to one another. So that's the stages of mitosis, or of the cell cycle, with mitosis technically just being only the PMAT part. Okay? So normally in mitosis in the cell cycle is controlled. There are a lot of measures in the cell that control this process. And there are checkpoints along the way. Okay? So we didn't talk about all these specific steps, and that's okay. But I want you to think of it kind of like your washing machine. So in order for the washing machine, before it starts spinning, it has to put water in, right? And it won't start spinning until the water is to a certain level. And then it'll start spinning, and, before you can open the door, the water has to drain. And so each of these things represent a checkpoint to make sure the things before it have actually happened. And that helps to control the cell cycle. So some of the checkpoints, like for example, this checkpoint at the end of interphase, G2 is at the end of interphase, that has to make sure all the DNA is copied. Okay, and that's important because we want the DNA to be fully replicated before we start to divide the cell. There's also checkpoints in metaphase. In metaphase, the checkpoint is to make sure that all of the spindle fibers are attached at the centromere. And so that makes sure that the chromosomes are split evenly and everybody gets an equal full amount of the chromosomes. Okay? And so these are checkpoints that normally happen to make sure that mitosis happens correctly. Sometimes, however, cells lose the ability to meet these checkpoints and they lose these controls that happen. And the result of that is that dreaded C word, cancer. Okay? So cancer cells have a few different properties. First of all, normal cells have contact inhibition. So this is a plate of normal cells. So you see little pockets of cells here. And if you put those cells in a petri dish with nutrient agar, which is like cell food, and allowed them to divide, they would divide until they filled the dish in a nice thin layer, and they would stop. They exhibit contact inhibition, which means that crowded cells stop dividing. When they form that nice thin layer, they stop. And that's what our normal tissues do, okay? Cancer cells don't do that, though. This is a cancer cell, and that is not contact inhibition. They just kind of continue to grow on top of one another. Normal cells, if you scooped a little piece out of the middle, like shown here, it would replace it and stop growing. Cancer cells, though you see here, don't do that. They kind of tend to mound one another, layer on top of layer. Looking at the shapes and stuff in this diagram compared to this diagram also doesn't look the same. Cancer cells also don't have anchorage dependence. So anchorage dependence means that divided cells must be attached to a substrate. They must have something to anchor them or hold them down. Another layer of cells, something else. But normal cells do that. Cancer cells don't do that. And so that's why they can break loose and grow in other places. This is another image of contact inhibition. Okay? And so here is a cancerous tumor. This one is starting in the breast, right? And then eventually part of this cell is going to break off and enter the bloodstream. Now we know that the blood goes everywhere in the body. And so it can then adhere to another location, and instead of just being localized in the breast, that cancer tissue has metastasized, which means to spread to a new location. Once it gets in the lymph or in the blood, it can travel to new spots and cause tumors elsewhere. Okay? That's why it's important, women, when you get to a certain age, they'll ask you to do annual, self-breast exams to feel for tumors, to feel for knots or bumps or lumps. And if you catch it early and you get it before it's metastasized, they can take the cancer out of the breast, or worse, they could take the whole breast. But both of those would be a better alternative than allowing and having the cancer to metastasize. So catching cancer early is really important to being able to treat it, okay? So not all tumors will break off. Tumors that break off and spread to new locations, as I've just explained in this diagram, are called malignant tumors. Benign tumors generally grow a little bit slower, and they stay confined to one area. Generally, you could go in and remove those and not cause significant problems. While I've had and know many people that have had malignant tumors that metastasized and moved to new locations, my best friend in high school, or in high school too, but in 6th grade, she had a benign tumor that was growing on the bone in her ankle. And so today, she has about a 4-inch scar on her ankle where they went in, cut it off the bone, and it was no problem because it was a benign tumor. It didn't cause any serious issues. It didn't move. It was slow and just, grew some extra cells over time. But those two are very different. So I want to now take a look at your page. Hopefully, if you haven't already, this will help you to be able to complete this paragraph here. And then you'll need to read about a pathologist and then just tell us a little bit about a pathologist here. But let's go ahead and complete this somewhat together, looking at cell division from a normal cell and a cancerous cell. Now this is on my PLTW, but I'll also just go through it with you. So in a normal cell, cells divide in a very organized, if I can write here, organized and my pen is struggling with me. It never has quite such problems. A very organized and controlled fashion, right? It stops when it's necessary, okay? But in a cancerous cell, it is indefinite. They just keep dividing and keep dividing. It's uncontrolled. Uncontrolled. If y'all can bear with me in my pen here. Okay. The size and the shape of cancer cells are very irregular. And hopefully you saw that in the one picture here. You know, we've got these little bitty cells here, and we've got these big, huge cells here. It's very irregular size and shape, okay? Versus a normal cell, they're small and they're round. They're also pretty uniform in size and shape, okay? In a cancer cell, the nucleus could have very large, irregular shaped nucleus, versus a normal cell has a small, round nucleus, okay? And there's just one of them. Sometimes these could be multinucleated. Specialized features. So those specialized features, like what I talked to you about, how the cell controls itself, they have cell control. They have features that allow for that. And then cancer cells lack cell control and the features that cause that. I'm so sorry I can't write or stay on my page. And then the last thing, arrangement. So in a normal cell, they are arranged in layers. Okay, they're in nice layers. They're flat. But in a cancerous cell, they are very irregular. Grow on top of each other. They break off. All of those types of things that we discussed. Okay? So hopefully this was a little bit more informative than just going through the stuff on PLTW, and you learned a little bit more about cancer today. You all have a great day.